Welcome to Dr. B.R. Ambedkar Open University. Good afternoon one and all. Myself Dr. Anitha, Faculty, Department of Zoology, Dr. B.R. Ambedkar Open University, delighted to be a moderator for today's teleconference on protozoan diseases. This particular teleconference actually has been allotted for the BSc students. It is as well important for even for the PG students. Now, to discuss about this particular topic, we have with us two resource persons. One is Dr. Meenakshi, who is an assistant professor from Department of Zoology, Nizam College, Osmania University. Madam has about 20 years of more than 20 years of teaching experience, and also she has published so many papers in research journals. And another resource person is Dr. G. Sridhar. He is also a faculty from Department of Zoology, Nizam College, Osmania University. Now, this particular topic, that is today's topic on protozoan diseases, is very interesting and highly informative. Now, as we all know, the protozoa are minute structures, and they are the first organisms to have occurred on earth. Now, these protozoa actually cause a number of dreadful diseases like malaria, leishmaniasis, amoebiasis, giardiasis, and so many other dreadful diseases affecting millions of people worldwide. So, to know about this particular disease, to know about this parasitic infection, especially the protozoan infections, it is very essential not only to overcome these diseases, but also to have awareness as well as to improve the public health. Now, these diseases, so many variety of diseases are there, and these diseases actually pose a threat. So, it is highly essential. Now, since we have these two resource persons, our resource persons will deal in detail about these diseases. Now, over to Meenakshi madam. Now, before going to Meenakshi madam, I would just like to tell that this program actually is for 1 hour, and dear students, if you have any queries, now you can dial to this particular number, that is 040-236-80456. Once again I repeat, 040-236-80456, and this will be available only at the end of the session. Now, I would like to once again welcome Meenakshi madam as well as Sridhar sir to discuss about this particular topic, and also to tell about this particular topics. Now, first I would like to tell Meenakshi madam. Now, Meenakshi madam, will you just introduce about the various protozoan diseases and also throw a light on the parasitism?
Okay. Thank you, Dr. Anitha, for inviting me for this program. As we all know, that protozoans, they are the first single-celled organism. Though they are single cell, but still they are a whole animal in itself. They contain all the organelles what a particular individual should have, and those organelles will function all the functions of an individual higher vertebrate, like respiration, digestion, excretion, stimulation, circulation. All these are being carried out by a single cell. So, they are very fascinating animals. They have a well defined nucleus. The nucleus may be one, or they can be two, or they can be multinucleated animals. Externally, these protozoans are covered with a plasma membrane, and they have the locomotory organs. They are motile. So, some of them are sessile, but majorly they have a locomotion. They move from one place to another place in the means of gliding or sliding, or by means of flagellar movement. So, locomotory organs plays an important role in their classification also. Now, the locomotory organs are flagella, they have the lobopodia, that is just blunt extension of the body, and cilia. Based on these three locomotory organs, they have been classified into different classes. One is the rhizopoda, that is lobosa. in which we have entamoeba and our normal amoeba. Now, coming to the ciliates, we have paramecium, that one beautiful example of the ciliates which we have already studied in our intermediate. Now, coming to the third one, that is the flagellates, we have studied about euglena. Apart from euglena, there are a variety of other flagellates present in that phylum, in that particular class. Coming to the last one, that is the sporozoa, this is exclusively parasitic. No doubt all the other classes also have free living organisms along with the parasitic organism. What are those parasitic organisms, which we will be throwing light on it? When we talk about the sporozoans, malaria, we might have already heard about it, which sir will be discussing about what malarial parasite is. Now let us see, when the organisms are free, how can they become a parasite? Paranna Jivulu, what we say it in Telugu, how they are going to become a parasite. Parasite is that particular organism which enter into a body of some other individual and stay there and derive its nutrition. So, initially it started with a symbiotic relationship. They gel together, they move together and they have a common association, and slowly the symbiosis has entered into a parasitism. The free living organisms, by chance, by virtue of some incidents, they might have entered into the food, and through the food it has been ingested by the organism. Once they entered into the organism, they have seen that they got a very good habitat and they started settling there because they were getting the ready-made nutrition. So, they started adapting to that particular environment, and based on that approach, they slowly turned into a parasitic organism. And this, during the parasitic nature evolution, we have seen that the symbiotic relationship what they had, it has turned into parasite. What does it mean? It started taking its nutrition, it started surviving in the body of the animal, and then slowly it started destroying the animal. Once it started destroying the animal, it has become a complete parasite, because in parasitism we know that there will be only one side relationship, that one host will be having a disease condition or affected, and the parasite will be living very happily. Not only that, during that, how it was able to do it, the co-evolution has occurred during the evolutionary tree, from the single cell organism to multicellular organism. During the evolutionary pattern, we have seen that the parasite which was there, it evolved along with the animal source, and during this evolution they have lost their alimentary canal, they have lost their other organelles which are responsible, like respiration was becoming anaerobic, the lack of mitochondria become very less. The major organelles that were developed was the endoplasmic reticulum and the Golgi apparatus, where it has to take the food, package the food, and then use the food for their survival. And most important aspect of that, it is that they had a multiplicative cycle. The reproduction has increased, so they have developed the multiplicative cycle in such a way to perpetuate their life, because once they entered into an human being or a particular animal or into a host, they have to come out and then spread the disease, or they have to get a new host, because once a host dies, they also will die. So, they had a very good life cycle wherein they started producing large number of eggs, or they had the situation where they can have a cyst so that they can live in the environment. So, that is how they has developed the process of parasitism. And one doubt we might be having, like when they are present in the body of an individual, we all have studied about body has an immune system or an immune mechanism, how they are able to tolerate. This parasite is very, very intelligent. As soon as it enters into the body of a particular host, it will localize in one particular cell, that is intracellular mobility will be there. So, our body's immune system will not be able to recognize them. Secondly, the antigenic variants, every time they come into the body, they have a different variance of the antigen. They show their different expression. Therefore, the body will not be able to accept it, and they have a molecular mimicry. That means the molecules that are present in our body or in the host body will be mimicked by them, so that our immune system will not be able to recognize it properly, and that is how it is able to manage the escape mechanisms, and they overcome all this. And slowly, as they were taken by the ingestion, they are locally placed initially in the alimentary canal, but from there it was only the fecal matter through which it entered into the environment. So therefore they slowly started migrating, they started destructing the tissues, and slowly the migration occurred, and it was very easy for them to be perpetuated or transmitted from the blood vascular system. So therefore they became a blood parasitic system. So parasitism, what we say is either intestinal parasites or they are a hemoparasite. They are present in the blood so that they can be easily transmitted, and therefore the transmission has taken a different route. It was not a single route of transmission. We had a second route of transmission with vectors. So vectors play an important role in the transmission of this particular diseases, and there are variety of causes for this transmission of these protozoan diseases. These protozoan diseases, the causes is poor hygienic conditions, or then we have not good habits of eating food and drinking water, contaminated drinking water, overcrowding. All these are certain risk factors and causes which lead into the protozoan diseases.
Thank you, ma'am. Now, Sridhar sir, will you discuss about the epidemiology and statistical data on these protozoan diseases?
Thank you, madam, for introducing me and giving good opportunity to explain about the protozoan diseases, because especially we are living in a subtropical country, especially mainly according to the WHO, the epidemiology of this protozoan diseases. If you see that nowadays we are highly developed, but though maybe we are having highly developed scenarios, but if you see that the statistics of the what is that these protozoan diseases, mainly malaria and amoebiasis, giardiasis, trichomoniasis, then sleeping sickness, you can also call it as trypanosomiasis. They are prevalently observing present day scenarios. For example, if you take the malaria, more or less according to the 2024 statistics, you can, if you see that 2.3 million people are suffering with this disease, mainly due to the different types of problems, mainly what is that drug resistance, poor hygienic conditions, and illiterate conditions leading to these problems. That is why discussion about the protozoan diseases for a degree level as well as postgraduate level, it is very important. If you see that amoebiasis, it looks like very simple disease, but more or less 2000 to 3000 individuals are getting fatal or getting mortality due to this only in India. That means out of the world population, amoebiasis more or less 30% of the individuals are dying in India. That is why, like dysentery, like amoebiasis, like that. That is why we can discuss very prominently about the protozoan diseases in the earlier stages of life science education.
Okay, thank you, sir. Meenakshi madam, we have got so many protozoan diseases are there. So can you explain about the pathogenicity, its transmission, what are the symptoms as well as the preventive measures about amoebiasis, giardiasis as well as leishmaniasis?
Okay, thank you, ma'am. Now we all know that we have just studied about what parasitism is. Now these parasites, they are causing lot of threat to the life, as sir has said. The epidemics, what we have seen, that they have been endemic to some of the regions and they were epidemic in some regions. Like for example, when we are talking about certain diseases like giardiasis, it is endemic to the areas of Africa, US, etc. So based on the poor hygienic conditions. So diseases are endemic or they can be epidemic. Now classifying the organism based on the disease condition, their localizing habitat, we have intestinal parasites and parasites. As I have already told you, intestinal we have taken for our discussion as two examples. One is Entamoeba histolytica and Giardia. The blood parasites we have taken as the Leishmania, then Trypanosomia, malarial parasite, etc. and so on. So now I will be focusing on Entamoeba histolytica, because the major disease that we see is the amoebic dysentery. Wherever we go and have our food, immediately we complain about stomach discomfort, the distension or the bloating of the stomach. These are the situations of might be the viral infection, or it can be bacterial infection, or it can be a protozoan infection. Generally protozoal infection will be feco-oral route only. That means from feces to oral route it will be passed on, as these organisms, they shed down their cysts into the fecal matter. When we talk about Entamoeba histolytica, it belongs to the class Lobosa, and Lambill and Losch in 1795 they have identified this particular disease, and later on they have given the complete structure and pathogenicity of this. When you talk about the structure of this, it is a blunt organism and it does not have a proper shape. We generally take it as a irregular shaped organism with one single protuberance, which is called as a lobopodia, one single lobopodia through which it will be moving around, and it is represented in two types, that is, one is in the trophozoite state and the second one is in the cyst state. And trophozoite is a growing and a feeding stage. It will feed on the material that is present in the intestine, it will feed on the lipids, it will feed on the proteins that are there, and mostly it feeds on the blood, so that we can see the ingested blood cells, RBC cells, in the structure of the trophozoite. And it has the cytoplasm. As we know that protozoa has a nucleus and the cytoplasm and the plasma membrane, it is covered by the plasma membrane. The nucleus here is very prominent. Here the nucleus will have a hollow, and in the center we have a nucleosome, and from there will be lectin radiations will be there, which will give like to a wheel-like appearance, which is called as a cartwheel appearance of the nucleus. Based on this becomes a diagnostic feature when we are identifying this organism. And apart from that, some globules like fatty globules can also be seen in the parasite. And this trophozoite, it is active feeding and growing stage as said. Once it enters into the body of the host, this parasite will be feeding and causing the destruction, and when it has to get out of the body, it will convert into a cyst form. When it is converting into a cyst form, the lobopodia is retracted and the size will be reduced. You can see chromatin bodies as well as the globules in the first stage, and slowly as it is becoming a complete cyst enclosing with a cyst wall, there these chromatide bodies and the globules will be lost, and it becomes a tetranucleate cyst which will enter into the environment. Now coming to the life cycle as such, as I told you now, this cyst, they are coming out through the fecal matter from the infected person. No doubt mostly all of us may be the carriers of this without any symptoms, but only the symptoms can occur when the multiplication of this organism occurs in the intestine. So those person who is having the large number of these load of these protozoans will see the symptoms. Now when it enters the cyst along with the food, contaminated water, it will enter into our body through the alimentary canal, reaches the stomach. In the stomach, the acid that is present try to dissolve these cysts and it go and localize in the intestine. When it is localizing in the intestine, the proteolizing enzymes that is there, they will be bringing about the destruction of the cyst there, and the cyst wall is being broken out, and there we see that excistation, the organism come out of the cyst, which is called as excistation, and this will convert into a trophozoite. Now when the trophozoite is there, this trophozoite will have two types of action. One is the non-invasive action, the other one is the invasive action. In the non-invasive action, what this trophozoite do is, it will just be present on the surface of the mucous membrane, which is present in the intestine, internal lining of the mucous membrane, and it will be taking the feeding from that mucous surface only, and slowly after a particular period of time it will convert into a cyst, and along with the other fecal matter, it will be just passed on into the feces. The cyst formation will take place and it will be passed on into the fecal matter. Other invasive is that it will not keep quite just attaching to the surface of that, but it start destroying the membrane of the intestine. It will start feeding on the cell, the columnar cells that are there, it will start feeding on it and leads into the death of that particular cell, and that death place will become a flask shaped funnel like structures, and those are called as ulcers. So flask shaped ulcers are being formed. When the ulcers are being formed, they become pleuritic. They are filled with yellowish brown color fluid into it, and because of the cell damage, it has the chance to damage the blood vessels also, and slowly it tries to escape from the blood vessel to various other organs, and this pleuritic ulcers that are there, they start secreting the secretions. Along with the stool, we will be seeing the blood as well as the black or brownish color tinge onto the stool. And the other part of that, when they are migrating to the blood, it will be entering into the various other organs, and this is called as the extra-intestinal migration, or it is the metastatic lesions. The metastatic lesions are the secondary lesions. The primary lesions are ulcer formations in the intestine, and then passing of the stools and stomach pain, etc. will be there. Secondary lesions is that it will enter into via the blood circulation into lung, it will be entering into liver, it will be entering, sometimes it may even enter into brain. So they cause damage to the liver, they cause damage to the lungs. When they enter into lung damage occurs, the breathing problems will occur. Liver, the destruction of the liver cells will lead into liver abscesses, necrotic liver, and it may lead into jaundice type of condition. These are the secondary lesions that we can see in Entamoeba histolytica, and majorly the symptoms what we can see is the abdominal pain, fever, intermittent fever will be there, and then we'll have the constipation type of symptoms, and anemic conditions will be there. So these are certain things, and complete loose motions with blood. The stools will be laden with blood, and when we see this, we have to approach a doctor and go for a diagnostic test. The diagnostics is by doing the microscopic analysis, take the fecal matter and observe it by staining with a Giemsa stain, identify it as a trophozoites or the cysts that are there. We have to identify that, and then we should say that whether the amoebiasis is there or not. And most important factor here is when we take the stools, the stool will be having a chocolate colored dots, which we are called as chocolate ligand crystals, which will say that it is infection of the amoeba. And then we can go for the treatment, taking medicines or the drugs like metronidazole or tinidazole, etc. So these are the drugs which can take, and most important is we have to avoid, because this destruction what it is causing in the intestine leads to lactose intolerance. Therefore milk products have to be completely banned, and then unsafe drinking water, we have to boil the water and drink, so that the contamination further can be reduced. This is about Entamoeba. Similar type of dysentery can be seen with Giardia. Giardia, it belongs to the flagellates, where the most important aspect of this is it is also an intestinal parasite, and it is caused by Giardiasis, is a disease which is caused by Giardia lamblia, and this is a racket shaped structure. It has 2 big nuclei and 8 flagella will be present, and there will be the rod-like structures which will be supporting this, and this organism will have a convex shape on one side and concave on the other side, so that when it enters into the body, it will go and stick to the wall of the intestine, and from there it start feeding onto it. And these animal, again, when we say it is having only one host, and Entamoeba also has only one host, that is directly from the fecal it will enter into the host and cause the disease. Similarly, Giardia also shows that it has only one host, where the infection will be causing to the host and then come out into the environment, and again there is no other mode of transmission for it. So again, this Giardia, the cyst of the Giardia will be passing on into the stool, and from the stool, fecal-oral route, again it will enter, and generally Giardia is present in soil, water, contaminated foods, etc. And there are great chances of contacting this giardiasis even with a sexual transmission also. There are certain cases, but usually it is through the drinking water, contaminated drinking water, and this is also known as a recreational disease. Why? Because mostly in the normal ponds, lakes, etc., that where we go for recreation, there the animal debris or animal fecal matter, etc., will be there. So therefore that contamination may enter into our body. So this cyst, when it enters into the body of a human being, or then it will undergo multiplication. When it is undergoing multiplication, it undergo multiplication by binary fission, and once this binary fission is occurring, the multitude of these organisms, they will stick on to the walls of the intestine and start feeding on the intestine, leading into abdominal pain, fever, nausea, vomiting like conditions, etc. And the stools that have been passed here, unlike the stool of amoebiasis, you will see that they are filled up with large amount of mucus and oil, and we can see that the mucus will be, the fecus will be so light that it will have a flotation, or it can float on water also. So that is an indication that there is a disease caused by Giardia. And the treatment, again, how do you identify this Giardia? Again, you have to go through the microscopic analysis, and it is very rare that you have to see, we can be identifying the cyst of this. So we have to go for antigenic tests like ELISA, etc. has to be done to identify these organisms, and then say that the suffering is with Giardia. And again, here it is, the treatment can be given by metronidazole, or it can be given with hydrochloride, etc. Then coming to the next type of the parasite. So these two were intestinal parasites where they were causing the dysentery. Coming to the third one, that is the Leishmania. Now the difference between these two is Leishmania requires another vector transmission. It is a digenetic parasite because it requires two hosts. One is the host where it is infecting, the other one is that what it is transmitting the disease. Now Leishmania, when we talk about it causes a disease called as Leishmaniasis. Leishmaniasis, again, it can generally we call it as a Kala Azar or a black fever, or it can also be called as a dum-dum fever, etc. Now this Leishmania, it occurs in two phases. One is the two stages, I can say it as. One is the amastigote stage and the other one is the promastigote stage. Amastigote, mastigote, masti means it is the flagella. Amastigote, it will not have a flagella. It will be a circular structure with a small basal granule and a nucleus. The cytoplasm is very well defined and it will be a round structure, and amastigote stage is present in human beings or the host, definitive host. Then coming to the promastigate, this promastigote, it has a long flagella, basal granule, and it has a nucleus, and it is spindle shaped in structure, and this is present in a vector. So the transmission of Leishmania's disease occur by means of a vector. Now vector here is sandfly, which is Leptomenas species, and when we talk about this Leptomenas, there are variety of species which are carrying these and causing different types of infections. Leishmaniasis is only various type of cutaneous or visceral or something which I will be dealing little later. Now coming to this parasite, when the cyst, that is the cyst that is there enters into the human being. It has been displayed on your screen. The amastigote form, it is oval shaped, and the promastigote form, it is having a big flagella, and that is present in the sandfly. This during the bite of the sandfly, that at that time the promastigote forms will be entering into the human body, and it will enter through via the oral route, and then it will enter into the neutrophil cells that are there, because first line of defense in our body is the neutrophils. Those neutrophils will engulf it, and then those flagellate cells that have entered, they will be releasing back, and the macrophagic cells that are there, because blood has monocytes, and these monocytes in different tissues will act as a macrophage. They eat up that particular tissue and they convert this promastigote into a leishmanial form. This leishmania undergoes division again, and multiplication will occur, and the macrophage will burst out, liberating the amastigote forms, and these amastigotes will be taken up by different macrophages, and those macrophages will be again multiplying, and the load of these mastigotes will be there, and slowly it will reach into the circulation. And when they are present in the circulation, because the neutrophils, macrophages, it's already ingested, and the number is so much that it is very difficult for them to engulf and destroy it. So they will be circulating in the circulation, and when this fly, sandfly, Leptomenas species come and feed on the blood, they carry this amastigotes into their body. It will enter into their stomach, and from there it will undergo the transition to form a that is flagellate form promastigote, and slowly it will come to the salivary gland, and from there when it is feeding onto the other individual along with the saliva, it will be entering into the next host. That is how the transmission of this leishmania will be occurring. And coming to the disease, it causes the leishmaniasis disease, and this leishmaniasis disease can be of three types, that is visceral leishmaniasis, cutaneous leishmaniasis, and the third one is the mucocutaneous leishmaniasis. Generally visceral leishmaniasis is present, and this is mostly a tropical subtropical region disease only, because in these areas it is most prevalent. And when it comes to the viscerally, it will be causing various organs will be affected. The spleen, most affected is the spleen, lymph, etc. These are the glands which will be affecting. It will go there and start destroying these cells, and they become totally impaired. The functioning will not be proper, and that is called as a visceral leishmaniasis. And because the function is occurring, and the loss of weight will occur, loss of appetite will be there, and it leads into drastic anemia and also darkening of skin. And because of the darkening of skin, it is also called as the Kala Azar disease. And then the other type, it is the cutaneous. Cutaneous, it is again caused by a different species like Tropioca. So cutaneous is only the skin layers. It will have an effect on it. It will form the sores on it. It starts eating the cell structure, and it will form a sore on it. Small boil like structures will be there. They are called as boils, Delhi boil sores or oriental sores. They will be large sores where they will be entering into them, they will be eating away the tissue, and the entire tissue will become filled with fluid as well as sores formation. Coming to mucocutaneous, it will enter into a deeper layer of it and destroy the mucus as well as the cutaneous layer, and mostly they will be affecting the nose membrane, the mucosal membrane of the nose, ears, mouth, etc. So it will lead into the disease condition of the entire, where it will form ball like structures. Small boils will be formed, and they will be hardened boils. They are called as macular boils. They are not papular, but they become hardened boils. They will not be having any pus, but they will be eruptions onto the body, which will give a different type of a look of that particular patient. Again, the treatment, what we need to do is aminopterin B is a particular drug that has to be given for the treatment of leishmaniasis disease. Thank you.
Yes. Thank you, madam. Now, next coming to other diseases like malaria and trypanosomiasis. Sridhar sir, will you please explain about these two diseases?
Okay, madam. Thank you. As madam told that some protozoan parasites are intestinal parasites, some protozoan parasites are infectious parasites, but mainly the trypanosomiasis and malaria diseases are mainly due to the blood sucking parasites. I mean to say that there are also both trypanosomiasis as well as malaria parasites due to the digenetic parasite. You can also call it as vector-borne diseases. The first we will discuss about the trypanosomiasis. Trypanosomiasis mainly you can also call it as sleeping sickness. Why it is called as sleeping sickness? Because the parasite will enter into the brain and causing the sleeping sickness. The main causative factor or causative parasite for this trypanosomiasis, that is called as Trypanosoma brucei and Trypanosoma cruzi. The species is different, but the parasite is same. Trypanosoma brucei mainly causing the African trypanosomiasis. You can also call it as a sleeping sickness. Then the next parasite in the trypanosomiasis, that is called as Trypanosoma cruzi, which is giving a disease that is called as Chagas disease. You can also call it as American trypanosomiasis. If you see the trypanosomiasis vector, there are different types of vectors in the trypanosomiasis. Mainly the sleeping sickness caused by a vector that is called as CC fly. Because of this bite of the CC fly, the parasitic stages will enter into the human circulation and causing the disease. First, mainly if you see the life cycle of this trypanosomiasis, if you sleeping sickness, the CC5 bite leading to the entry of parasite into the human circulatory system. The causative factor for this CC fly disease, what is that? That is called as metacyclic tropomastigote. This metacyclic tropomastigote will enters into the circulatory system. After entering into the circulatory system, first it will enters into the blood fluids. Then after entering into the blood fluid, it will undergoes to the continuous binary fusion or asexual reproduction. This continuous binary fusion, it will go on increasing the number. This metacyclic tropomastigote, this tropomastigote converted as a tropomastigote. This tropomastigate now will enters into the blood circulation to lymph fluid. This is a parallel circulatory system which we will find in our body. From lymph fluid to it will enters into the brain, but while entering into the brain, you need to cross the blood brain barrier. The only one protozoan parasite which can cross the blood brain barrier, that is trypanosoma. After crossing the blood brain barrier, it will enters into the brain, not only into the brain, through the brain it will enters into the spinal fluid. After entering into the spinal fluid, which will cause the sleeping sickness, there also it will undergoes to the continuous binary fusion and form a stumped structure. Stumped structure means it will be very big and small. It looks like a stump. Then this stumped structure you will find in the what is that body fluid like blood, lymph as well as spinal fluid. Then after entering into the spinal fluid, then it will enters into the next life cycle, that is in the CC fly. If there is a bite with the CC fly, this stumped what is the tropomastigote will enters into the CC fly. After entering into the CC fly, once again in the gut of the CC fly, it will continue its life cycle. This tropomastigate, after entering into the CC fly, it will converts as epimastigote. This epimastigote, once again it will goes to the longitudinal binary fusion, then increase the number. Due to the longitudinal binary fusion, this epimastigote is converted as procyclic, what is that tropomastigote, procyclic tropomastigote to, once again it is converted as epimastigote. Now this epimastigote will enters into the salivary glands of the CC fly. Then after entering into the salivary glands of the CC fly, once again it will continue the binary fusion and converts as metacyclic tropomastigote. Now this tropomastigote once again will enters into the what is that human being. Like this it will continue the two life cycles, one life cycle in the human being, other life cycle in the what is that in the CC fly. Okay, this is about the sleeping sickness which mainly causing in the tropical areas and Southeast Asia like India and mainly Africa and North America. This is the main causative disease in the tropozoan parasites and blood sucking parasites. And what is the main treatment for this disease? If you see that this what are the problems which clinical symptoms we will find for the bite of the what is that CC fly. You can see that the diagram, you can see what is that you will find a wound formation, because the CC fly is not just like mosquito, just because mosquito will just inject the blood and suck the blood. But if you see the what is that CC fly will damage the tissue on the skin, and which will leads to the what is that wound formation in the human beings. If you see the clinical symptoms of the what is that this trypanosomiasis, mainly it is causing the fever, severe headache, irritability and extreme what is that tiredness, swollen lymph nodes and what is that paining of the different types of muscles and what is that bones. Then what are the main treatment which you will take for this disease? There are the different types of combinations this disease, but the main combinations to overcome this sleeping sickness, that is suramin with pentamide, because generally what we will say that suramin combinations. Suramin combination means the main drug here suramin combined with the different types of drugs like pentamidine, then what is that tyripos. These are the main what is that treatments which you can take for this sleeping sickness. Then come to the next parasitic or blood-borne protozoan disease, that is malaria disease. Malaria disease is one of the most common disease in the protozoan disease and one of the major disease in the what is that tropical diseases which we are finding what is that in addition with the Wuchereria disease or filariasis. Yes, what is the main causative parasite for this malaria disease? The main causative parasite, that is Plasmodium. Plasmodium is a sporozoan. As madam told that at the time of classification, because all sporozoans, mainly sporozoans in the what is that in the classification of protozoa, they are belongs to parasitic in nature. Yes, while discussing about the plasmodium, there are mainly 4 types of plasmodium species we will find, that is Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, Plasmodium falciparum. Out of this Plasmodium vivax, ovale, malaria and falciparum, this falciparum is a highly fatal condition. That means if you are getting the what is that this Plasmodium what is the falciparum infection, maximum it leads to the death. That's why we will consider this is as very fatal in condition. Then if you see the what is that where exactly we will find the problem, mainly the Plasmodium vivax enters into the liver tissue or hepatic tissues, as well as what is that RBC or erythrocytes, what we are finding in our body. And what is the transmitting factor here? Because as I madam told that there are some vector borne diseases in the protozoan diseases. Just now we have discussed that trypanosomias, there is a vector that is called as CC fly. Here in malaria disease, the vector, main vector is that is what is that Anopheles mosquito. This Anopheles mosquito, mainly that what is that female Anopheles mosquito bite is leading to the malaria fever. Then if you see the what is that life cycle of the malaria or Plasmodium vivax, this Plasmodium vivax mainly having the different types of life cycles, as mentioned to the digenetic parasite. Digenetic parasites means the life cycle it will continue in the both in the human being as well as what is in the mosquito. Especially if you observe in the different other parasites, generally asexual what is that life cycle observed in the vector, sexual life cycle observed in the human being, but contradictory to this other parasites. But in Plasmodium, what is that parasite? Plasmodium parasite will do the asexual life cycle in the human being, sexual life cycle in the malaria, what is that mosquito. If you see that in human being also, stages we will find the different types of stages. The first stage, it will enters into the human being as a sporozoid stage by the mosquito bite. After entering into the human being, it will converts as what is that sporozoid is converted as merozoid in the liver. Then from the liver it will enters into the RBC. In the RBC it will converts as what is that gametes by a process that is called as a sporogony. Then that gametes will enters into complete the asexual life cycle in the human being. Then it will enters into the what is that once again into the mosquito. After entering into the mosquito, it will complete its sexual life cycle and form the sporozoid once again. Like this, it will converts. It will continue its life cycle continuously in both human being as well as in the mosquito. If you observe here in the slide, the digenetic parasite mainly in man, it will convert as asexual life cycle in 2 phases. The first phase that is called as what is that in the liver, you can also call it as liver schizogony. The second phase in the human being, that is called as what is that erythrocytic or RBC schizogony. That means it will enter first, it will enters into the liver, then it will converts into sporozoid stage to cryptozoid or merozoid stage. Then in this merozoid stage, it will stop, or it will sometimes it will convert, it will re-enters into the liver, sometimes it will enters into the RBC. After entering into the RBC, it will continue and form the especially gametes. Then you see that mosquito, after entering into the mosquito, it will show its sexual life cycle, then fertilization will occur in between the gametes and form the zygote as oocyst. Then due to the sporogony, it will form the what is that sporozoid. Then what is the life cycle? First we will see, this is the sporozoid stage, which is the main infective stage to the human being. It is a spindle shaped structure. Here we won't find any flagella like what is that zoomastigophora, what we are finding like the giardia and all these things, but here you won't find any what is that flagella. Then it is having a single nucleus, mitochondria, all these things like a cellular organism we will find this structure. Then this structure or this sporozoid stage, it enters into the human being through the bite of the mosquito, then it enters into the bloodstream. After 24 hours to 72 hours, it will enters into the hepatic tissue or hepatic cells, or like what is that?
Yes, sorry to disturb. Yeah. Yes, continue.
Hepatic cell or hepatic cell. After entering into the hepatic cell, it will take that this hepatic tissue and eat the hepatic tissue and going to the what is that binary fusion, that is that form called as Sijont.
Hello? Hello?
Hello?
Yes?
I have one doubt, madam.
Yeah, you can ask later. 1 second. Yes, sir.
Yes, this in hepatic tissue it will undergoes to the multiple fusion. That multiple fusion what we are finding in both RBC as well as in hepatic cell, that is called as schizogony. Schizogony, after undergoing to the schizogony, this what is that sporozoite is converted as cryptozoite. Cryptozoite converted as cryptomerozoite. This cryptomerozoite now enters into the what is that liver cells as well as RBC cells. If you want to continue the hepatic cycle only, it will re-enters into the liver tissue, continuously eating the what is that liver tissue and damaging the liver tissue. Then after that, the stage that is called as pre-erythrocytic phase, it will enters into the exoerythrocytic phase. After entering into the exoerythrocytic phase or continuous liver stage, we can call it as it will form the cryptomerozoite to metacryptomerozoite, and what is that this metacryptomerozoites also once again two types. What is that micro metacryptomerozoites and mega metacryptomerozoites. Now the important concept here, generally mega, that means large merozoites will re-enters into the liver cycle, then small merozoites will enters into the erythrocytic cycle. Erythrocytic cycle means, that means RBC cycle. That means once these merozoites are micro metacryptomerozoites entering into the RBC, you can see here invading the RBC, that means it will leach the RBC plasma membrane and entering into the RBC. After entering into the RBC, this merozoite is converted as tropozoite. That means you see sporozoite in the liver cell is converted as merozoite. Merozoite after entering into the RBC, it will convert as tropozoite. Now the tropozoite having the different types of stages in the RBC.
Sir, will you conclude?
Okay. Tropozoite is converted by eating the what is that RBC. The tropozoite stage is converted as a singlet ring stage. In the tropozoite stage, it will form a vacuole-like structure. Slowly, when time is going on, the vacuoles, what is that space will increase. The what is that? It looks like a ring. What we are finding, what we are wear to the what is that finger. Then it will convert as amoeboid stage. In the amoeboid stage, we will find the small granule-like structure due to the continuous invasion of the RBC. These granules we can call it as Schaffner's granule. Then last stage we can find that Sijont stage. In this Sijont stage, the number of the tropozoites will increase in the RBC. Then the RBC will be burst. When RBC is increasing, bursting, when it is growing in the RBC, it will eat the hemoglobin and cleave the hemoglobin and form the hemojoin-like structure. This hemojoin is the main factor which will increase the body temperature, That is called as, what is, you can say that what is that erythrocytic schizogony. Now in the Sijont, after Sijont stage, it will convert, it will form the gametes. These gametes will re-enters into the what is that into mosquito, what is that female mosquito. Then into the female mosquito, after entering into the female mosquito, fertilization will occur. This fertilization leads to the formation of what is that oocyst. This oocyst from the gut of what is that mosquito re-enters into the salivary glands of the what is that mosquito. After entering into the salivary glands of the mosquito, this oocyst is converted as ookinete. This ookinete is generally, oocyst is not non-motile. Ookinete is a motile form. This ookinete is once again converts as what is that sporogony and form the sporozoites. These sporozoites once again entering into the what is that human being after completing his sexual cycle. You can see the sexual cycle, both all the cycles, human being sexual cycles as well as asexual cycles in the diagram. Then what is the main treatment for this disease? The main treatment, you can say that mainly quinone based structure. Mainly quinone based structures, and you can say that to decrease the body temperature, because sometimes we will go to the 106 Fahrenheit degree centigrade, we can say we can take that paracetamol. These are the drugs which we can take and overcome the problems due to the cause of malaria fever. Then what is the pathogenesis of this malaria? The fever also in different stages. You will find the cold stage, hot stage and sweating stage. In cold stage you will find the shivering at the time of hemogyne releasing. In a hot stage it will increase the, due to the what is that hemogyne, it will increase the body temperature up to 106 Fahrenheit. Then sweating stage, after 1 to 4 hours of the sweat, you will find what is that hot stage, you will find the sweat, total body is sweat and it will be cooled. This is about the main disease about the malaria. Thank you, madam.
Thank you, sir. Question. Okay. Now you have talked about the various diseases like malaria, trypanosomiasis, amoebiasis, giardiasis, leishmaniasis. Okay, can you talk a little bit on the management, means how to manage these protozoan diseases?
Yes, as just now the starting we have discussed that nowadays we are in 2025, everything is highly developed, but especially in subtropical countries and tropical countries, due to the main population, the first problem population, and the second problem, what is that? Poor hygiene. We are not following anything, the food intake, everything, this is hygienic conditions. Then third thing, drug resistance. That the drugs what we are discussing about the protozoan diseases, generally that chloroquine, metronidazole, that all these drugs are what is that very old 1930s and 1940s. That's why first we need to what is that educate the public about the population increasement, because mainly Asian countries and African countries are the highly prone to this protozoan disease. First we need to educate, you need to maintain the what is that these hygiene conditions, then we can decrease. For example, you say that amoebiasis, in 1994 the number of amoebiasis more or less 1 lakh, due to the highly education from the different types of what is that disease centers as well as different types of organizations like NGO, WHO countries, what that they are educating you, please what is that overcome these problems, then the number of amoebiasis disease decreased in India mainly like 1250. That means the number decreasing from thousands to hundreds. Then coming to the drug resistance, that means for a small fever we are taking the paracetamol. I mean to say that give your immune system some time to overcome or to develop the antibodies against the disease. Don't take azithromycin, don't take paracetamol, but give some time to your immune system, act against the parasite, act against the antigens against which is present on the parasite. Then this is about the drug resistance. Then third one, mainly the mortality rate is increasing due to the diagnosis. Mainly the diagnosis, if you find, the diagnosis mainly microscopic diagnosis, protozoan diseases maximum microscopic diagnosis. Nowadays they are developing the what is that molecular based diagnostics, that means PCR based diagnosis. If you are finding some problems, don't give any medicine to him, give some paracetamol and don't give the direct antibiotics, to don't increase the drug resistance. Take some blood sample from them and do the PCR. Okay, I mean I know that it is cost effective, but it will decrease the drug resistance in the concept. That means molecular diagnostics tools, increasement of molecular diagnostics tools is very important for us. Then fourth important thing, nowadays the kits are developing. For example, in as Madam told that microscopic what is that investigation, but we can also do here what is that kit based methods. There is a kit for malaria. The main base, main base for that kit, that is called as LDH, lactate dehydrogenase. That means the lactate dehydrogenase which we are finding in the plasmodium, what is that Anopheles, then we can overcome this problem. These are the different types of management methods. What is that decrease the what is that drug resistance, maintaining the hygienic conditions, then educating. Then the last one, vectors, mainly CC fly and what is that mosquitoes. These are the vector borne diseases. If you stop the vector transmission or decrease the population of the vectors 100%, we can decrease the what is that progression of these protozoan diseases.
So LDH, that means you talked about lactate dehydrogenase, which is an enzyme. Does it act as a marker?
Yes, it is an active marker. What they will do, they will purify that protein and they will keep that in that kit and Western. That is a just basic Western blot paper. They will keep that protein in the Western blot. Then you take your blood sample and separate the serum. It is very easy to separate the serum. Then take some serum sample and add the serum sample by adding some buffer. Then you can detect as COVID kit.
As COVID kit.
As
So
COVID
increase
kit.
or decrease based
Yes,
on that you will
yes, ma'am.
infer the
Yes.
result.
At least we will find that antigen is there or not.
Okay.
Okay.
So this is the the latest technology that has
Latest
been developed
technology
for
developed in the
malaria.
malaria.
That we can do it in our labs.
Yes, you can do, because take that kit, because the kit cost is more or less what is that 100 to 50 to 100.
Okay.
Okay, then you can buy, take some serum sample and add the serum sample. Okay, then you can check it, because you are having the control line and what is the test line. If you are having the line only at control line, that is negative. If you are having the two lines in the kit, that is control line and test line, that is positive.
Yeah, this test works very good.
Very good and
Easy,
what is it?
quick.
Easily, quick. That means then we can decrease the mortality rate of the disease.
Otherwise, based on microscopic method means it takes a longer time.
100%, because you see that to check the trophozoite stage
Yeah,
in the blood sample,
yeah,
more or less it will take 7 days time,
yeah.
or 3 to 4 days time. In that 3 to 4 days time, maximum damage will occur in your liver tissue as well as RBC.
Okay. So based on the just like COVID, you are saying this is a kit method. So just based on the kit, you will come to know whether that antigen is there or not.
Yes.
So still any further modern technology is there or modern tests are there to detect? Like for that basic test, for as we do it for COVID, we go for the basic test, then they go for PCR and all those things.
Yes, PCR
What about
test.
in this technique?
PCR technique also you can do, because mainly what is that you can take the blood sample, isolate the DNA and do the PCR by using the what is that plasmodium specific antibodies.
Okay.
These are primers. Then you can do the PCR. Okay, in that PCR, based upon the after run the gel, then after getting the PCR, then you will find that it is positive or negative.
Okay.
Okay.
Well explained, sir. Students, if you have any queries, you can ask the questions. Yeah. Further, any information you have regarding any other protozoan diseases? Of
Yes,
course, these are the some of the diseases you have talked about, because these are the most common diseases as well as the dreadful diseases we talk about daily in our lives. What about the other protozoan diseases?
There are some other protozoan diseases like trichomonas.
Okay.
Or you can also call it as a trichomonas vaginalis. Okay. This is sexually transmitted disease. Just now we have discussed that intestinal parasites.
Yeah.
Then what is that vector-borne diseases. Now this is a different disease which is caused by the protozoa, that is called protozoan parasite, that is called trichomonas vaginalis. Okay. You see it is pear-shaped structure. You can see the slide, but pear-shaped structure. Okay. It is also having like mastigate or flagellum. Then it is having the undulating membrane, but the undulating membrane which is different with the trypanosoma, because in trypanosoma you will find undulating membrane all over the body, but in this parasite you will find only half of the body.
Yeah.
Then what is that the transmission of this parasite mainly that is sexual intercourse. That's why you can call it as this trichomoniasis, like sexually transmitted disease. Then you said here also that infective stage is the trophozoite stage, mainly the urethra and vagina. Mainly it is found in female urethra and vagina. Then in male it is found in the urethra as well as prostate gland. Okay. After introduction of this sexual intercourse, proliferation begins with results to the inflammation, a large number of trophozoites present in the vaginal fluid and causing the fever and infertility problems in the human being. Okay. These are the other prominent sexually transmitted disease and protozoan disease which we will find in the human being.
There will be a green color discharge also. So with that we can identify that it is trichomonas, and it will have a foul smell. A typical type of a smell will be there, and the sticky secretion will be secreted.
Thank you, sir. So lot of research actually has been taking place, still going on. But till now regarding malaria, we are going, it is highly impossible for us to defeat these mosquitoes, because every day it is giving lot of menace. So till now, but no one has actually come forward with a vaccine. Why? What is the main reason behind it?
Yes, coming to the vaccines for this parasitic diseases, because for example, if you take the virus or some bacterial disease, we will target one protein.
Yeah. Okay,
one protein which is very prominent in that particular bacteria or virus. But we are coming to the malaria disease. This is not a single single causative factor.
Okay.
There are different types of Plasmodium species, because we will discuss only 4. Nowadays in India we are finding one more what is that Plasmodium species, that is called as Plasmodium novalis.
Okay.
I mean to say that while we are increasing the technology, at the same time the parasite also increasing its own resistance.
Okay.
That's why there is very less chance to develop the what is that vaccine.
Okay, thank you, sir. Thank you, Meenakshi madam, and thank you, Sridhar sir, for your enlightening and very informative discussion. Thank you once again.
Thank you.
Thank you.
Thank you.